The video is one of my favorite videos of what's going on inside of a cell, and it's just got enough zen. There's a link to the video, and there's a link to the video of a longer version of the video with narration that tells you what's actually going on in there. But at any rate, we're going to be talking about the molecules of cells, and all of the things that you saw, all the interactions you saw in that video were interactions of molecules, because nothing in a body, nothing in any kind of living creature doesn't happens without there being an interaction of some kind of a molecule, of some kind of a charge. Otherwise, nothing happens. There's no hand waving going on. Um, So the first thing we're going to talk about is a little bit. We're going to talk about organic compounds, and organic in this context is different from organic in the grocery store, right? Organic compounds here means carbon based, and then the compounds we're going to be talking about is carbohydrates, lipids, proteins, and nucleic acids. And we'll just talk about them individually just a little bit, and then we'll talk about why they're important. So organic compound are carbon based. Carbon, we saw before on the shell that there's four outer electrons, so it needs four more, so it can bind to four hydrogen ions, for example, hydrogen atoms, for example. Um, and so these are very typical configurations. What can happen? These are called hydrocarbons. So you've got carbons over here that are surrounded by hydrogens. And when you look at the names, you've got a propane over here, a butane. You may recognize this as fuel, right? Propane is what you use in your gas. Um, um, in your propane tanks, right? To when you're grilling. And then you've got butane, that's lighter fluid. So these are things that contain energy, and the energy is contained in the CH bond. So these are, as I said before, hydrocarbons. And so it should be a very small step to carbohydrates right from there, right? Um, the same kind of deal. Um, but back for one more second here. So these are energy containing items. The other thing, so this is the basics of the carbon based structure. The other part that I don't usually use that much in the beginning courses, so I'm just going to mention it, and we you can forget it immediately right afterwards. um, Most of the reactions depend on functional groups. So if you see a hydroxyl group someplace, an OH, then it'll probably react at some point, potentially with a carbonyl group. Um, One of the things, the carboxyl group and the amino group, you're going to see those again. Those are part of the peptide bonds. They react with each other. The phosphate group is important for energy. And then the different locations of methyl groups change the function of a molecule completely, or can change the function completely. For example, here you've got a methyl group that's added here in testosterone. You take that away and make a couple of other modifications, and all of a sudden your male hormone becomes a female hormone. So these small little details make all the difference in the world. And so this is why this it's important for you guys to know that. When some charlatan out there says, "Oh, this molecule looks very much like this other molecule, so therefore it's equally poisonous," That's usually complete nonsense. Um, but The ones we're going to be talking about are macromolecules. Macromolecules usually are made out of individual little parts. Macro means big, right? So they're large molecules, and most of macromolecules are made out of monomers. Mer means it's just a part. Monomer is like it's a one part. Oligomer, it's a few parts, and polymer is many. Poly means many, so that's many of those parts. So macromolecules usually consist of polymers of whatever monomer. Um, the way those polymers are formed is usually very similar in each of the cases. In most cases, it's a dehydration reaction. So water is the glue that keeps this whole thing together. Um, and so when you remove the water, that's when you stick them together. When you put the water back in, that's what separates them. And these reactions, they're mediated by enzymes that are again proteins themselves. So this is a visualization of this. So you've got a short polymer that has the hydroxyl group, and then you've got the hydrogen free over here. So you remove the water that binds these things together. If you add the water back, you can take them back apart. And that's literally what happens with every single one of the polymers that I put together. So you've got glucose and fructose here, and what I want you to see is the carbon chain and the hydrogen ions coming hydrogen atoms coming right off of that. Um, and that's your energy, right? There's energy contained in this, and we all know that there's energy in glucose, right? Um, there's another way of writing the formula. They can be circular, or they can be straight. And here is that reaction again, this polymerization where you've got the hydroxy group and the hydrogen, the water is removed. These two are bound together into maltose. You do that a bunch of different times, and you end up with a polymer, either starch, glycogen, or cellulose. Now, starch is what you find in potatoes. That's the storage that plants use. Glycogen is the storage that we use in our tissues, and then cellulose is structural for plants. When you look at these molecules, they're all made of glucose monomers. They're all polymers of glucose. But when you look at the where the oxygen is, these are all directed in the same direction, right? The bond is all identical bonds in both the starch and the glycogen, whereas here you've got alternating sides where the bond is on alternating sides. We can digest this. We have the enzymes that can break this down. We don't have the enzymes to break this down. So, what a body can do depends on the enzymes that are contained in the body, and so that's a very important thing. Um, when we fight different creatures, for example. Another macromolecule is lipids. Lipids, you've usually got the glycerol and you've got the fatty acids, and they're tied together with that same dehydration synthesis. And you've got carbons over here, a long chain of carbons and hydrogen to either side. This is nonpolar, meaning that there's no charge anywhere. It's completely not positively or negatively charged. Um, and so that's a fatty acid. Now, when you have the complete fatty acid, usually looks like this. You've got like you've got the glycerol, and you've got like three fatty acids um, attached to it. And this one is a saturated fatty acid, and this one is an unsaturated fatty acid. The difference between the two is that one of them is completely straight and the other one has a bend in it. This is important because it actually tells you where you can find saturated and unsaturated fats. If something's really straight, you can pack it very tightly and you can make it into a solid. So the saturated ones that don't have the bend in it, those are going to be your butter and your saturated fats that are solid fats. Whereas the ones that have the bends in them, the unsaturated fats, those are the crooked ones. You can't pack them as tightly. So that those are the unsaturated fats. Those are in the oils. That's how you can remember that. A very specific and special kind of phosphate lipid is phospholipids. Life without phospholipids just literally would not exist. What's magic about phospholipids is that you've got this hydrophobic tail over here because there's no polarity to this at all. And then you've got this hydrophilic head, and because of that hydrophilic head and the hydrophobic tails, when you put this in a soup, all the hydrophobic tails are trying to get together, and all the little hydrophilic heads are trying to get together. And they make these little things called micelles. They kind of look like the eyes on a soup, like the fatty eyes on the soup. You'd see them getting together where all the little hydrophilic heads would stick on the outside and all the little feet would be on the inside. Or another configuration that works is a bilayer where all the little hydrophobic tails are together in two layers, and then the hydrophilic heads are together exposed to the water. The moment you have that, then you have two compartments. And the moment you have two compartments, you can have things happening in one compartment that is not happening in the other compartment. And the moment you have compartments, you have the potential for life. So without compartments, that's almost impossible to do. Um, Proteins are the other macromolecule cool we got to talk about. Um, the shape of molecules is very, very important, and so this is a stick configuration of what one of those molecules may look like. This same molecule in a space filling model would be like that, and whatever this molecule does, it may be able to bind like right in here, because like I said, everything in molecules has to do with shape. um, Molecule spider webs are made out of molecules, proteins. Proteins are, yeah, they're amazing, what all they can be. They're basically made up of amino acids. So they're a polymer of amino acids. Amino acids are these things. There's an amino group and a carboxyl group and a carboxyl group and a rest. The rest is, those are different molecules, but structurally all amino acids look like this. And then when you have several amino acids, so the rests, those are the different kinds of rests that you can have. And they have different characteristics. Sometimes they're hydrophobic, sometimes they're hydrophilic, and especially the hydrophobic ones, they kind of tend to stick together, make little hydrophobic pockets away from all the water around them. Um, and when several amino acids get together, you again have this dehydration reaction where these two are removed as water, and you've got a peptide bond. Once you have bunches of peptide bonds, you can make a very long chain of amino acids that all look sort of like that, with peptide bonds between them. And this long chain of amino acids would be then the primary structure of a protein. This folds immediately based on whatever little charges, little hydrophilic interactions there are, um, into different kind of regular structures. Some of them are called alpha helix. Some of them are called beta pleated sheets. Um, those are secondary structures. Those are just structures that are secondary to just the chain of amino acids. The tertiary structure then is the final structure of the protein that's stabilized by the secondary structures, additional R groups, additional interactions between all the different parts. Some proteins are functional at that point, as in that tertiary structure. In some cases, multiple proteins have to get together in order to become a functional protein. That last one is then called the quaternary structure. One of the things I guess I want to point out here is as you're reading through the book, don't discount the pictures. You see me talking basically through the pictures. Um, Pictures in biology books are not just there for illustration. They're there for you to look at it basically almost like as a summary. And then if you don't understand the summary, go back into the text and look at the text from there. Last but not least, and we're going to do more about nucleic acids later, but we're just going to skim them right real quick here. So we've got DNA, deoxyribonucleic acids, and RNA. DNA is a double helix. We've got DNA. RNA is a single polynucleotide chain. And all of them serve as the blueprint of who we are. They're set. They look about the same. For the DNA, for example, you've got the deoxyribose and nitrogenous base and a phosphate group, which they get together and make this sugar phosphate backbone with a nucleotide. There's 4 different nucleotides: adenosine, thymine, cytosine, and guanine. And in the case of DNA, you have this double helix um, where the T goes with the A and the C goes with the G. Um, when it comes to what used to be called the the dogma, um, from you start with DNA, it gets transcribed into RNA, and that gets translated in protein, and that this is the only direction it can happen in for human beings. So we've heard a lot about RNA right lately in terms of vaccination, right? So RNA literally is a protein that's not a protein, is a nucleic acid strand that's made for the short term to just, not to go back into the DNA or change anything in the DNA, but for the short term to just be translated into protein that then's recognized by antibodies, for example. Um, and that's all I wanted to talk about for this chapter.